Overview of analytes, proteins. So the basic structure of proteins are polymers or macromolecules that are built from one or more unbranched chains of amino acids. There's a primary, secondary, tertiary, and quaternary structure to proteins, and that's depicted over here. So primary structure is simply the order of amino acids in the polypeptide chain. We call it a polypeptide chain. Poly means many. Peptide refers to the type of bonds that exist between adjacent amino acids. So polypeptide means many peptide bonds. We're joining many amino acids together. Secondary structure has to do with the hydrogen bonds that form between adjacent amino acids to produce a kind of two-dimensional folding. So we have the most common would be alpha helix or beta sheets. So alpha helix provides like a helical shape, beta sheets is folding like this, and then we also have some random coils that can exist in our secondary structure. Tertiary structure has to do with the hydrophobic interactions that fold this tertiary structure into a three-dimensional structure. And then when we get to quaternary structure, we have multiple tertiary structures coming together to produce our final protein. So general chemical properties, they contain many ionizable groups on the side chains of their amino acids as well as on their N-terminal and C-terminal ends. The pH of the environment determines the structure of that protein. So at certain pHs, the side chains are going to want to interact or not interact, so they're going to expose themselves or hide themselves depending on the pH of the environment. The pH at which an amino acid or protein has no net charge is known as its isoelectric point. We can do isoelectric focusing in order to either identify or separate out particular proteins. Synthesis. So predominantly they're synthesized in the liver and then secreted into circulation. As a review, protein synthesis goes through the process of transcription and then translation. There's also post-translational modification where the protein is modified into its final form. Protein catabolism. So when we consume protein in the diet, our digestive tract is going to secrete digestive enzymes, which are going to cleave the bonds, the peptide bonds of adjacent amino acids. Once those are cleaved, we have the peptide, and then we have a free amino acid. That amino acid is then going to undergo deamination. Deamination removes the nitrogen component, creating ammonia. And then there's an organic acid that can either be recycled to produce more amino acids, or it goes into the process of glycolysis or the Krebs cycle. So lots of fates for this guy. Ammonia, though, is a waste product. It's toxic, and it must be converted to urea in order to be eliminated in the urine. So ammonia gets converted to ammonium, and then eventually through the urea cycle, it gets converted to urea. Urea is produced in the liver and eliminated in the urine. Plasma proteins are the most frequently analyzed of all proteins. There's two main protein groups, albumin and globulins. Globulins consist of the alpha-1, alpha-2, beta, and gamma fractions. The predominant lab tests that you'll see, so if you order like a basic metabolic panel, typically they want to evaluate protein levels, and that's going to help determine liver function and all sorts of other stuff. We evaluate total protein, albumin, globulins, and then the albumin to globulin ratio, also known as the A to G ratio. Plasma protein values are listed here, so you can see the reference values and some more details about each one of these. Prealbumin and albumin. So prealbumin, or transthyretin, is so named because it migrates before albumin in the classic serum protein electrophoresis. It's a transport protein for the thyroid hormones thyroxine and triiodothyronine, T3 and T4. Low concentration may also indicate poor nutritional status. Albumin is a negative acute phase reactant, meaning that it decreases in the presence of acute disease states such as inflammation, infection, or injury. Albumin is responsible for nearly 80% of the colloid osmotic pressure. In other words, that helps maintain the osmotic pressure of the bloodstream. That has a direct correlation to blood pressure. If we have protein in the bloodstream, then we're going to have water in the bloodstream, and we're going to maintain a good blood pressure. Increased renal loss of albumin, termed albuminuria, commonly occurs in renal disease. Globulins. There are four main types or fractions of the globulins that are designated as alpha 1, alpha 2, beta, and gamma based on their electrophoretic mobilities. So when we put these in a serum protein electrophoresis, the different fractions are going to migrate at different rates. And that's how we categorize these globulins. Examples are listed below, and we're going to go through each one of these fractions. So reference ranges for albumin, the globulins, are listed in this table. The alpha 1 fraction contains alpha 1 antitrypsin, phytoprotein, acid glycoprotein, anti-chymotrypsin, inter-alpha trypsin inhibitor, and the GC globulin. The alpha 2 fraction predominantly consists of haptoglobin, ceruloplasmin, and alpha 2 macroglobulin. The beta fraction is pretty extensive, so you can read through this list: transferrin, hemopexin, lipoproteins, microglobulin, complement, fibrinogen, C-reactive protein, and high sensitivity C-reactive protein. And then the immunoglobulins. These are our, in the gamma fraction, these are our antibodies: IgM, D, G, E, and A. Serum and plasma are acceptable for measuring for protein. Total protein is lower at birth, but reaches adult levels by 3 years of age. The Biuret reaction is widely used in the determination of total protein concentrations. In an alkaline medium, in the presence of at least 2 peptide bonds, cupric ions will complex with the groups involved in the peptide bond. Then we use a photometric assay in order to determine the concentration of protein. Albumin is most widely used methods for determining our dye binding procedures using Bromkressel green or Bromkressel purple. So fractionation, identification, and quantification. So salt fractionation uses sodium salt to cause precipitation of globulins, and that leaves behind albumin. Albumin, again, we do that dye binding by electrostatic forces, and then that can be directly measured. Total globulins, the measurement in serum is by direct colorimetric methods using glyoxylic acid. Albumin can then be calculated by subtracting globulin from the total protein. And then we have the electrophoresis method. In serum protein electrophoresis, serum samples are applied close to the cathode end of the support medium that is saturated with an alkaline buffer. Support medium is connected to 2 electrodes, and current is passed through the medium to separate the proteins. Protein fractions are fixed by immersing support medium in an acid solution to denature and immobilize the proteins. Proteins are stained, and then they appear as bands on the support medium. The membrane is inspected visually or with a scanning densitometer. The most significant finding is monoclonal immunoglobulin. So here you can see a typical pattern of what we would like to see in a densitometer. So just to kind of show you what this is versus what this is the actual gel. So we load our samples into here, and then we're going to turn on the current. The samples are going to migrate through the gel based on size and charge, and then we will see a separation of the bands. Then we're going to use what's known as a densitometer. So the densitometer is going to determine the density of the bands or the protein concentration within the different bands and produce a graph for us. So the higher the peak, the more dense the color in the band. So you can see in a normal one what the pattern would look like, and then you can start to see abnormalities. So an alpha-1 antitrypsin deficiency, you would have a decrease in this area. If you have inflammation, you would have increases in these areas. Here's monoclonal increase, so all of a sudden we have a great deal more of the gamma fraction. That means we have a lot of antibodies being produced. Nephrotic syndrome, you can see this pattern, and then cirrhosis is indicated here. So different patterns can indicate different disease states. Capillary electrophoresis is similar to serum protein electrophoresis. It's just instead of taking place on a membrane, it takes place in a capillary tube. So the use of capillaries allows heat to be effectively dissipated, which means higher operating voltages can be used, and that improves the separation and analysis. Immunofixation electrophoresis also is commonly used. So what we do in immunofixation electrophoresis, it allows us to identify the proteins using antibodies. So we run through the typical electrophoresis, and then we use antibodies to help stain the membranes, and we can evaluate them that way. High-resolution protein electrophoresis allows for further separation of proteins into as many as 12 bands. So instead of seeing the typical 5 bands, we can separate it up into 12 bands. It uses higher voltage cooling system and more concentrated buffer. This allows us to determine individual components within each one of the main bands. And the main bands I'm referring to are back here. If we go the albumin globulin, alpha-1, alpha-2, beta, and gamma globulins. So we can see separation within those bands. And then isoelectric focusing is a different method. That's zone electrophoresis that separates proteins on the basis of their PI, so their isoelectric point, the point at which they have a zero charge. Other proteins of clinical significance, troponin. So there's cardiac troponin CTN, which represents a complex of regulatory proteins. Troponin C, TNC, or troponin I, CTNI, and troponin T, CTNT, that are specific to cardiac muscle. CTN measurement above the decision limit along with clinical evidence is indicative of myocardial injury. In other words, a heart attack. Natriuretic peptides is a family of structurally related hormones that include atrial natriuretic peptide, ANP, B-type or brain-type natriuretic peptide, C-type natriuretic peptide, and dendropsis natriuretic peptide. NT-proBNP and BNP are found in greatest concentration in left ventricular myocardium. Fibronectin. Proteins are found in plasma and on cell surfaces and can be synthesized by hepatocytes or liver cells, endothelial cells, peritoneal macrophages, and fibroblasts. Fetal fibronectin is used to help predict short-term risk of premature delivery. Cystatin C. Cystatin C may be used to evaluate glomerular function in individuals for whom creatinine measurements may be misleading. Hemoglobin. So this is classified as a transport protein. Its role in oxygen transport from the lungs to the tissues, and it transports carbon dioxide back to the lungs. Hemoglobin synthesis occurs in immature red blood cells, and it's located in the bone marrow. or immature red blood cells that are located in the bone marrow produce the hemoglobin, and then they're sent out into circulation. So hemoglobin is definitely measured to determine anemia. And then myoglobin is the primary oxygen-carrying protein found in striated skeletal and cardiac muscle, and that accounts for approximately 2% of total muscle protein. Myoglobin can be elevated in the diseases that are listed in the table to the right. So myoglobin used to be one of the major indicators of a heart attack. And you can see other conditions that it's related to.